We great. Welcome everyone to our first Thursday speaker series webinar. It's a November 7th, 2024, and a little after 7. And tonight's guest speaker is Jay Holmes, and I'm going to introduce him in a little bit for you. But first, I would like to thank the people who help us bring these speaker series to the fore. Lou Siegel, who has been a great supporter and also the person who does all of the making of the flyers and the announcement of the flyers, the promotion of them on our different platforms. So thank you, Lou. And I also want to thank Russell for his IT support, which he helps us out with every month. And before, during, and even after the series is over. We do this series monthly, and we record them and we have them for our members to be able to access them on our website. Just a few little announcements. Our organization is in the middle of a new visioning strategic planning process. So we hope that you, our audience, will respond in any way to any appeals that we will put out during this process to get your input. And we very much want to bring you along with us and have your input be a part of this process. Our upcoming holiday party is on Saturday, December 7th at Alley Pond Environmental Center at 4 o'clock. It'll go from 4 to 8, and so we hope to see you there. And we have been sending out announcements in our Strandline newsletter and on our website, and we will also send it out on our Facebook page. Unless I've forgotten anything, if anybody from the board wants to jump in and add any reminders, if not, I'll then proceed to introducing Jay. Okay, Jay and I have known each other for, I don't know, 30 years, something like that, at the Museum of Natural History. And we both worked in the education department. He's still working there, and I'm retired. But he's currently the senior manager of the Urban Advantage Professional Development at the AMNH. And the Urban Advantage is a partnership between 8 science institutions in New York City. Jay develops and facilitates courses for teachers in earth science, ecology, evolution, and the nature of science. He joined in 1992 as a lecturer for global warming, understanding the forecast, and has been a coordinator of the high school after-school program, advisor for the Youth Can Environmental Conference for Youth, the AMNH Ecology Club for Teens, and he developed and implemented hands-on activities in the Discovery Room before joining the Urban Advantage team in 2004. Jay was president of the New York Microscopical Society from 2019 to 2023 and enjoys exploring and sharing the smaller nooks and crannies of New York and the cool creatures that live there. And I was speaking with, I think, the president of the Microscopical Society 2 nights ago and exploring other ways we can have their involvement for our first Thursday speaker series. So without any further ado, I'd like to introduce Jay, and Jay is going to talk to us about Darwin and the HMS Challenger and anything else he considers important.
I think.
Go ahead, Jay.
Okay. Hello, everybody. Thank you, Lisa, for that intro. And I think we'll just dive right into the slides, and I'll explain a couple of things along the way about this, the reason for tonight's gathering. So I'm going to start sharing screen. And if people have questions, just jump in.
Well, we have a Q&A for questions, Jay. After your talk, it flows a lot better that way, and this way we
Okay,
can focus on what people are asking about.
sure. And I always have trouble keeping track of the chat, so
Okay. Yep.
Um,
Sure.
sorry, now I got to find my slides. Oh my goodness. Here we go. Whoop. And I'm gonna go like that. So we're going to explore a little bit about microscopes that have been used on voyages of various sorts over the span of about 80 years, I guess, or no, maybe closer to 40, and depends on how far we get. We might get right up until the 1970s. Um, so this presentation sort of came out of a post that I made on Facebook that Lisa spotted. I play around with some old microscopes and slides and stuff, and have done a little bit of work around the history of microscopes. And I had posted this on this little story on Instagram that ported it over to Facebook, and it was about a microscope and a slide and a book and a little story about those three things. And that's what we're gonna get into today. My interest in microscopes started way back when I was in elementary school. My sister got this microscope, and this was a Bausch and Lomb, and she had a whole kit. And I managed to find just one of the scopes on eBay and pick it up and play around with it once in a while as a historic tool from my life. I, over the years have always sort of kept an interest in microscopes and ran across this book by Brian J. Ford called The Single Lens. And it's about one of the kinds of microscopes that we're about to look through, maybe on the table here. And it was connected to a story that was shared here at the Museum of Natural History. We had a Darwin exhibit, and we had one of Darwin's microscopes on display as part of the exhibit. And so this really got me going into Darwin's use of microscopes. And over time, when he went on this voyage, he was like 22, and he got a little advice from some of his professors about what kinds of tools to take along on this voyage. And if you look at this plan of the Beagle, uh, you'll notice in the back here, there's a little room with a couple of people sitting at a table and some bookshelves behind their head. This is the space where Darwin spent 5 years. And he was on this voyage as an extra. Normally on voyages during this period, the ship was a naval ship or some kind of a merchant ship, and there would be a surgeon, a doctor on board, and they often had the role of being the naturalist and collecting specimens or recording observations of organisms and stuff that were encountered during the voyage. And on this particular voyage of the Beagle, Captain Fitzroy was very interested in having like a full-time naturalist and somebody that he could talk to about natural history. And the word had been floating around for a little bit, and things weren't panning out. And one of Darwin's professors ran across this interest and shared it with Darwin and thought that he might be a good candidate for this experience. And it took a bit of negotiation between one of Darwin's uncles and his dad to get permission to go on this voyage. But eventually it happened. And he ended up in this space. And this space was not a cabin, it was the chart room, the poop cabin, where the captain would spread out his charts on this big table and navigate. And so Darwin ends up in this space. And you can see down at the bottom here the scale in feet. So this thing is like 1, 2, 3, 4, 5, 6 feet long. So if a bunch of us lay down, we would probably come to about there, if we were laying flat out. And this table was about 6 feet by 4 feet, something like that, and took up most of the room. And this big thing right here is one of the masts. And these things sticking out here are controllers that control the rudder. So as you're walking around this room, you're stepping over all this stuff and around all these things. So there's not a lot of space. And Darwin ends up hanging from a hammock that is hung over the table, and that's his bed. And there's penciled in, this is a copy that Darwin had, there's penciled in descriptions of what these things are, and I just transcribed them. So chest of drawers, washstand, instruments. This is where Darwin kept his microscope. And the microscope that he had was a single lens microscope, a little very portable thing, and actually a little large as far as single lens microscopes went at the time. And we'll take a look at that in a second. When Darwin went on this voyage, again, he was just sort of stuffed in this chart room and had to bring all of his own equipment. And he brought oyster trawl frame, just the opening, the metal piece that goes on the opening of a net. And he used some wool bunting that was on board the ship. Wool bunting is sort of a light wool fabric, but it wouldn't, and the way it was woven, it wouldn't stretch and distort when it got wet. So he made this net out of wool and cast it over the side. This thing was something like 4 feet long and like 2 feet wide, something like that. And this is a sketch from his diary on the Beagle. And he would leave this thing towing behind the boat during the day, and then he would spend his evenings looking through his microscope at what he had collected. And one particular thing he described as, I never saw anything more beautifully luminous than this coralline was. When rubbed in the dark, every fiber might be traced by a blue light. So this was a bioluminescent organism. Corallines we call hydroids nowadays. And this was his sketch in his field journal on the Beagle. And it's a little sort of branching colonial structure. And it has these little, they look like little wine glasses, transparent, sort of rigid structures with a little polyp inside. And this is the main stem, and these little branches with their polyps come off of these main stems. And you can access these drawings through the University of Cambridge Archives website. See if this will load. There it goes. And you can see he would record his thoughts and things in one journal and do his illustrations on these other pieces of paper. And so this particular piece of paper has a whole bunch of drawings of different organisms. And you can get to some pretty high resolution versions of these things and zoom in on them. While I'm streaming video, it's pretty slow. Here it goes. And so you can see the kinds of drawings that Darwin was making in his field journals with his fountain pen back in the day, or quill. And so these are some of Darwin's actual illustrations. And I'll see if I can find my slides again. Here we go. Get back into presentation mode. Just go to the next one. So this is the tool. This is what he was using, and this is what I have on the table in front of me. This particular one was made in 1825, and Darwin's was made in about about 10 years later. And each one of these was custom. This is before mass production. So each one is a little different. And Darwin's had a little fine focus knob down here that slowly moved this column part up and down. And this particular focus knob moves the stage up and down. His also had an extra little arm that stuck out below the stage with like a little magnifying glass in there, and that helped focus the light from the mirror onto the specimen to get every drop of light out of the light source as he could. And this all fits in a box that's about the size of a cigar box. Let me see if I can stop sharing, and I'll go to what camera do I want to go to? Oh my goodness. Sorry, got a little small here. Okay, so what I have on the table is my cell phone on a tripod, and here's the little microscope. And see if I can zoom in a little bit. Get it in a position here. So I'll spin around this way, sideways. So here's the focus knob, and this particular kind of microscope is called an aquatic microscope, and it has some interesting features. There's a sort of dual stage that's on a little pivot here. And so you can swing the stage this way, and you can also rotate the lens. The lens is just in this little piece here on this arm. And so it kind of has a mechanical XY stage axis. You can swing these two things at about 90 degrees to each other and move the specimen around without disturbing it too much. And this glass stage is actually concave. It's dished, so it's really thin in the middle. And you would put an aquatic sample on that stage, and it would stay put in the center. And you could put a cover slip on it and move your lens and stage around to observe. And inside the box is a whole set of lenses. There are 8 different lenses here. And there's a thing called the live box, which is a little brass cylinder that unscrews, and it's got a glass top and bottom. And so you can put a little specimen in there and sandwich them in the box and put it under your microscope and see them live. There's another little stage that has a cork center, and it's wood. And this is for working with something that you might pin to the stage to work on. There's a pair of forceps and doing this one-handed, a little scalpel and probe. So it's a whole dissection kit and microscope in a box that's, about the size of a book. And so a very portable thing. And in the period, in the early, in the 1820s and up until the early 1830s, single lens microscopes actually produced sharper images than a compound light scope. You had problems with chromic distortion, and they hadn't quite gotten to the point where they were making lenses out of different colors of glass to correct for the chromic distortion stuff. They weren't using flint glass and crown glass to I'm trying to, whoops, not that camera. Now that sorry, just a second. We'll play around with that camera on the microscope again, see if we can get it to work. So they hadn't gotten to the point where they were able to stack lenses and correct for the color distortion that they got. And so those little rainbows that each lens makes, if you stack a whole bunch of lenses together, you just keep magnifying the rainbow and you get fuzzy boundaries, fuzzy edges. And so with a single lens, if it was a really nice, good lens, you could produce a much sharper visual experience than with these compound scopes. So this is the kind of microscope that went on a voyage in the 1830s. See if I can go back to my slides. Once we get through this whole story, we're going to try to look through some of these microscopes with my phone and see if we can get it to work. This is a coralline, a hydroid that was collected here locally in New York. These things grow on piers and stuff, rocks around the estuary, and so you can find them. And the image on the right is taken through this microscope, looking at this one of these hydroids. I have a couple different ones in jars here. And it's pretty sharp. You can see nice crisp edges on that thing. You can see little grains and grit inside there. I have another one that I'll try to get the image of that's more like the one Darwin saw, with the more like a wine glass sort of structure with a little polyp down inside. Let's see if we can do that. Anybody recognize this kind of thing?
The Daphnia?
It's like a Daphnia, it's a cladoceran. And this is in Darwin's journal from the Beagle voyage. So he was seeing some freshwater Daphnia and other things. And these are things that our students in class, might experience under the microscope. So it is kind of fun to have access to these Darwin illustrations and think about, oh, we're all observing the same stuff under these tools. And just a little quote about Darwin's use of microscopes. And this was a letter to his cousin, William Darwin Fox. And the pleasure of working with microscope ranked second to geology. My background's geology, so I can relate. I strongly advise, this is the way he wrote it, you to instanter to buy from Bancks in Bond Street a simple microscope such as it's the one that Mr. Brown of Brownian Motion, he had one of these microscopes too, and he recommended this to Darwin. And then take out some insects scientifically, by which I mean separate, examine, and describe the trophy. It's very easy and exceedingly interesting. I speak from experience, not in insects, but in most minute crustacea. So things like those little Daphnia, those cladocerans. So he was having a blast on this voyage. He's like 22, right? And he comes back and starts doing a lot of other stuff. And he gets another microscope because he starts studying barnacles. All barnacles that he can get his hands on, living and fossil. And he starts doing some really detailed investigation of their structures. And so he needs higher magnification. And for that, he gets a compound scope. And so this is in the 1840s. And the technology has come a long way in figuring out how to make compound microscopes, multiple lenses, and correct for color distortion and all that kind of stuff. And this is the one that he got, and this is in the museum in Cambridge now. And you can go check it out there. Fun trip. The Whipple has all kinds of science tool exhibitry, telescopes, a lot of optics. Whoops, sorry. Woo, went too far. And so it's an interesting visit if you're over in that direction. And these are the kinds of illustrations that he was making. Again, going to the Biodiversity Heritage Library. This is from one of the volumes he produced on barnacles. And you can see the kinds of structure on the limbs that he was recording in his microscopical observation. Let's see if we can zoom in and get this thing to load. Maybe. Oh, I didn't like that. Where are we? They're loading so slowly. So this is back on dry land, where you can have a big hunkin' microscope and and stabilize it and make some amazing observations. And he really spent 8 or 9 years looking at barnacles and trying to figure out the diversity in form and structure in this whole group and think about their relatedness. Okay. And he's doing that sort of stuff through microscopical observation. uh, he spends time with this compound scope, but his real love is actually these single lens, or the structure here is often called a dissection microscope or a botanical scope. And he approaches the guys who made this big compound microscope for him and has them build for him or make for him a really heavy duty microscope like this one we were just looking at. Only this one's a beast. It's got a really heavy column, and he had this big dish designed for it that would hold his barnacles in a fluid, in alcohol or some preservative, and he could dissect them in that dish. That was about a 3-inch diameter dish, and he had a whole bunch of different lenses for this thing. And this was a big oak base, and the company Smith and Beck eventually lists this design in their catalog and calls it the Darwin microscope. There are very few of these things around. This one was in a book that was written by a person who was deeply interested in these single lens microscopes and had a whole big collection, and he gave it to a museum in France. So Darwin moved on to plants after the barnacles. And this is where we start getting into the microscope that's on the Challenger. Darwin's illustrations, he doesn't do a ton of illustrations in his notebooks when he gets back on dry land. And this is one of his drawings of a carnivorous plant, a sundew, that he focused on. After the Origin came out, he dove into orchids and did a book about orchids, and then he did a book about carnivorous plants. And this was one of his illustrations from his notebook. And then he adds this little note in the book that the pictures in the book are actually drawn by his sons, Francis and George, because this is the kind of drawing that Darwin is doing at the time. And these are the drawings that his sons produce and get included into the book. So they were a little bit more detail oriented in their drawing abilities. And I work with students, and a lot of times I say, that's a scientific drawing. It represents, in a symbolic way, it's kind of a stick sundew. And this, is maybe a more artistic representation and more detailed, but, it kind of gets across the same point. He was talking about how the plant responds to a food item being placed on one side of the leaf, just half the leaf responds. And, that illustration captures it as well, to different degrees of accuracy. So Darwin is looking at these plants, and he's noticing some stuff going on in the tentacles, in the little branches on that leaf pad. And he needs to get closer. He needs higher magnification. And these are some drawings that he made with some higher magnification. And he's looking at what he's calling red globules moving around in the tentacles. And this is his science notebook drawing. And then if I stop sharing for a sec, this is the book, Insectivorous Plants. And let's see if I can get over here. I think I'm on this camera over here. Nope, I'm on this camera over here. And here are transcriptions of the exact illustrations. And if you took these two and put them side by side, is journal drawing and this representation in the book, they're exactly the same. And it's a little like animation sequence. Let me go back to sharing. Sorry, each time I pop out of the sharing thing, it goes to a little screen. Share. I'm gonna go to my slides. So this is a little cartoon animation kind of thing. This is drawing number 1. And you can see this big glob and this little matchstick down at the bottom. And then this is drawing number 2. The glob's a little bigger. The matchstick is a little longer. Number 3, the matchstick is really long, almost reaching the glob. 4, it hits the glob. 5, it sort of merges with the glob. And then another little bit sort of comes down in 6, and then things start to break up in 7. And so he was observing this stuff happening sort of in and between the cells. And he's doing that with this technique. He's taking one of the leaves, he's feeding it a little bit, and he puts it under a quarter objective. This note down here. In that day and age, they didn't write, this is my 10x objective. They wrote the focal length. So a quarter objective is a quarter inch. So it's got a focal length of a quarter inch. And he's noticing these little red globules moving around. And I was going to add a picture there. Never mind. And it's at this point that he gets this Hartnack microscope. Hartnack was a microscope maker who was in Paris at the time. And he right about this time, is moving to Potsdam. And so Darwin's son, Francis, the drawing person, had a Hartnack microscope for his university work. And he loans it to Darwin for a little while. And Darwin loves it. And so there's a big conversation going back and forth between Francis and Darwin about, let me see, I thought I had the link. Sorry. It's kind of a funny sequence, and you can go on, there's a website called the Darwin Correspondence Project, and you can type in Hartnack, and you can see all these letters between Darwin and Francis about the magnification and the tool. And he really wants this water immersion lens, which is just the breaking cutting edge technology of the time. And Hartnack makes some really good ones. And we nowadays have these oil immersion lenses where you put a little drop of oil between the cover slip and the objective lens, and it helps reduce light loss and glare. And so it helps with really high magnification lenses where you need every ounce of light you can get out of the situation. And so Darwin is trying to get one of these things, and Hartnack is in the process of moving to Potsdam, and it's taken forever. And he says, it's already been several months since I asked you kindly to send me a microscopic objective, of which I gave you the description. I ask if you can send it to me as soon as possible, since I would like to use it soon. This is Darwin writing to Hartnack in London. And this is an example of one of those microscopes. And it's the microscope that I posted on Facebook and that Lisa saw, and sort of started this conversation about microscopes on board ship. So the HMS Challenger was the first dedicated oceanographic trip. Darwin went on a trip as someone that the captain could talk to, sleeping in a hammock over the chart table, and hitching a ride for 5 years, and using a little corner of this space to store his instruments, his specimens, and, all of his clothing. He's really squeezed in. In 1873, the Challenger is a fully fitted ship. This is the Challenger at Gibraltar. And you can see a little smokestack in the middle here. It actually has steam power that it can use if it needs. But it's got 3 masts, and it traveled mostly by wind at the time and didn't use the steam all that much. They were out at sea for, 3 years. And when they came back, this was a dedicated scientific expedition with a whole suite of scientists, and they produced tons of volumes. These 3 comprise volume 18 of the whole set. And this picture was taken in the stacks at the Museum of Natural History. And the reports for the voyage take up 2 full shelves of part of the stacks. And this is the portion of the report that deals with radiolarans, little microscopic organisms. This is the vessel. I put this little bar down here to show the length of the Beagle. So the ship that Darwin went on was that long, that black bar, and the Challenger and Darwin was just in this little cubby in the back with all of his stuff. And this whole vessel was dedicated to science. And I'm going to zoom in a little bit. There was a whole naturalist workroom lab for handling the specimens, observing, preserving, all that kind of stuff. And over here is another science lab. This was the chemical lab. So they were doing analysis of the water along the way. And so they had these two dedicated spaces. And in these reports, there are illustrations of these spaces. And this is the naturalist lab. And I'm going to go to a website that has a zoomable copy of this. And we can zoom in. And there's a couple of microscopes here that have very distinctive shapes to them. This one over here has a triangular base on it and tilts back as two eyepieces. And this one here has this little sort of Y-shaped horseshoe base on it with this little arm holding the mirror. And it has this little other arm sticking off here. This is holding another lens. And these two are, let me see if I can do this again. I'm going to switch to this camera, see how that works. Boink. Okay. Zoom out a little. Okay. So this is a Beck. This or Smith and Beck or Beck. This one is Smith and Beck. Smith eventually leaves the company. He was the founder, and so it eventually becomes Beck and Beck. And so this is the one that was on the right, the little triangular base one. Smith and Beck is the same company that made the big tall compound scope that Darwin had. This one here. So these two are made by the same manufacturing team. And at this point, you know, things are a little bit more less customized and more production line kind of thing, but these things still are not being made in the thousands. And one thing that's really interesting about Smith and Beck is that they kept a delivery log and they recorded everybody that they sold these microscopes to. So you look on the base and you find the serial numbers on these things, and you can look up online. The microscopical club in the UK that scans all the delivery logs. And so you can go there and look up your serial number and see who the original buyer was. And so there's an entry for Darwin's big Smith and Beck. So on the Challenger, they had one of these guys. And they had one of these guys. This is the Hartnack. And you can see it's got this little, I'm going to move some things so it's a little less cluttered. It's got this little arm with the lens on it out here. It's got this little mirror hanging out on this little sort of arm on a swivel down on the bottom. And a very specific kind of look to it, very vertical. And it's got that Y-shaped base. And so when we see that in the drawing, go back to sharing. We can sort of, sorry, I'm in the wrong window, zoom, bring me back to zoom, the camera. Jeez, sorry. Share. Okay, there we go. So when we go back to the thing, we can see that there's that little mirror hanging out there and the little bullseye lens hanging on the side. And one other thing about this voyage, there's the nice picture of the scope. they were collecting all kinds of samples and had all kinds of tools dedicated to this because they weren't living out of a cabinet in the chart room. They had all kinds of gear. And they were doing all kinds of sampling. They traveled down around Africa, and then they went through the Indian Ocean, around Australia, up to Japan, then over towards Hawaii, and then down along South American coast, back around, back up into the Atlantic. So they went that way. And along the way, they recorded their locations and all kinds of sample gathering and sounding and all that kind of stuff. And this is a big dredge that they would drag along the bottom, and it had sort of a scoop at the open end of the net, and then it dragged these little tassels off the back end of it, and those would catch on stuff on the seafloor. And when it was brought up to the deck, they would untangle anything that was caught there, and they would empty the contents of the net on the deck. And then this is another kind of dredge or trawl that they were using when they were in deep water. And then when they were looking for small stuff, they were using these things, the sounding machine. And there was an original design they had on the ship, and there were some glitches with it. And so this particular design was actually designed by this person, Bailey, on the ship. And it has a bunch of weights, and it has this core, this hollow pipe in the middle of it, and a couple of perforations on the top here. And this thing, it said in the book that it was typically extended about 15 inches below the weights. And so this is kind of like a apple core. And they would drop it overboard. It would sink like a dart down to the sea floor, and then boom, stick into the mud on the bottom. And then they would release the weights. There was a little clasp up here that they could trigger that would release the weights, and then they could pull this whole thing back up, just the core with the mud stuck in this tube up to the surface. And there's a couple of things I'm not totally clear on exactly how many times they dropped this kind of device to the seafloor. In one place I was reading, they did 492 soundings, and they're calling this a sounding machine. But those could be just depth soundings and not sample collection soundings. And at another point, they mentioned doing 354 dredgings. And one person was writing like the contents of this tube were the dredgings, but those big nets are called dredges. So a little clarity required there, but somewhere between 350 and about 500 bottom samples were gathered during this voyage. And this is an image of all this gear sort of hanging together on deck. You've got a tow, like a plankton tow, and then here's the sounding machine, and here's that trawl or the dredge with the tassels on the bottom, and then the larger, simpler trawl that they were using in deeper water. And all this stuff would hang off of these are basically like bungee cord cages here. When the ship is pitching and moving around in the ocean, the rope would snap if the ship did a big turn. And so they hung these little pulleys from these like shock absorbers. And if the ship moved, these things would stretch and they wouldn't snap as they were dragging these trawls and dredges across the sea floor. So this is all apparatus for, gathering science samples. And this is a winch, a steam-powered winch on the deck that would help hoist the rope. And the process of lowering the rope took like an hour or I think an hour to go 1,000 fathoms. And some of their samples were down 5,000 fathoms. And it took about 3 hours to bring that 1,000 fathoms up. And they had to store all this rope on board. And after doing a sample, they had to dry it first before they could stow it. And so this must have been a crazy process. There are a few illustrations in the report. There's at the beginning of the report, there's about 3 volumes of narration that just sort of describe the tools and the process and the voyage. And then there's about 30 volumes of descriptions of like the radial area or foraminifera or things that they analyzed when they returned from the voyage and some analysis on board in those labs. So here you can see them emptying a dredge and the deep trawl that they were using and some sample jars, some glass sample jars on deck there as they're going about this. And they had a tow net, which is kind of like a plankton tow. And they would use that as they were moving along in this sort of horizontal mode. They would drag it behind the boat. Or when they were doing some of these deep soundings, they would drop it vertically down and have it like arranged with a weight on the back end of it. So it would come straight up through the water column. And they would use those samples. This is sampling jars, a couple of them here. And it seems like the scientists usually wore fancy hats and stuff and suits and ties. And the deckhands and stuff used other clothing. Some of the sediment that they brought up, they would sieve, and they had a variety of sieves with different mesh sizes. And this is an illustration of sifting out some sample in a basin and then having a series of jars that they would load the sieve contents into after sorting things a little bit. And in the report, they have an illustration of what typical samples looked like. There were zones that were rich in diatoms, and they were calling it diatom ooze and radiolaran ooze and globigerina, which is a type of foraminifera ooze. So as they went around the world, they were seeing that they're going to different depths and they're finding different kinds of sediment organism fragments in different zones. And so these are a few illustrations. And this is the chart, one of their charts. And up here at the top is Hawaii. They go down past Tahiti and then head east towards the coast of South America. And if we look a little closer, each one of these points is a sounding point. If there's a circle with a number, that's a sounding spot. And sometimes there's just a dot, like this one down here, there's just a dot in the middle. And so that's just a place where they were measuring current or wind direction, that kind of stuff. And the arrows represent the speed of the current, and the squiggly ones are surface currents, and the straight ones are current at some sort of depth, not too far down, but not at the surface. And you can see there's a little point where the current sort of flips around here. It says northern limit of countercurrent, southern limit of countercurrent. So in this band here, the current's going to the east, and above north of that and south of that, it's going to the west. So they're noticing things like that. And then these bold numbers are depth to the sea floor. And there's a couple of little abbreviations that represent the kind of sediment they found at that spot. So R.CL is a red clay, and rad Oz is radiolaran ooze. Yeah. And so when they come back, they've got all these jars of sediment, and somehow those things got distributed. And different microscope slide manufacturers were making slides of this sediment from the Challenger voyage. And this is one that I got and that I shared in that Facebook post. And you can see here, it gives the date August 26, 1875, longitude and latitude, and the depth in fathoms. And so if I go back to this chart, see this 2750, August 26 or 27, somewhere in here, 27, 26, it's all in this little area. So this is the location where that slide sample came from. And I purchased this on eBay. These things pop up on eBay fairly frequently. And, you know, I'm fairly confident that that's probably actually a sample from the Challenger. It's got the location and depth and all that kind of stuff fairly accurately. Um, and this is the image that I took and shared over Facebook with that of those radiolarans. This was a handheld shot with my cell phone through the lens, so it's a little fuzzy, but when you're looking at this thing through the microscope and can play with the focus on your own, it's amazing. It's really sharp. Um, and this was taken through the Hartnack microscope. And this is that radiolarin volume. Um, and uh, Ernst Haeckel was the illustrator for a bunch of these things. He's pretty famous for these sorts of illustrations. And they were looking at live stuff. This is sort of cytoplasm that's sort of extending out of the radiolarin. And so to get these illustrations, they must have been looking at live stuff under the microscope. And the whole book is just amazing. One of those volumes was the plates, that stack of 3 volumes. So one of them was just full of plates of all this radiolarin stuff. And the other 2 volumes were descriptions of the organisms. And they were finding new species of all kinds of stuff. There's fish, echinoderms, there's uh, everything. So lots of new species descriptions going on. And of course, they are on ship. And this is the base of that Hartnack microscope with a little holdfast, a little peg in a hole on the deck in the countertop that was clamping down the base of the microscope. Um, we're at 8 o'clock. How late can we go?
We're doing good. Keep going.
Okay. And so, as far as we're concerned, around our area, um, we don't need a deep sea vessel to go collect samples of the ocean floor, the sea floor. Some of you might be familiar with Big Brook in New Jersey, and it's a site that people go to. If many people go there are very interested in shark teeth, and that's like the big item there. You can go with a screen and a little trowel and just sift through the sediment in the bottom of Big Brook, and it's eroding these clay cliffs, these clay formations, and washing away the clay and leaving behind the big chunky stuff in the stream bed. And that includes shark teeth and other things. And I used to take high school students out there in the 90s, and I collected a little bit of the clay junk. And this was when we were working on the discovery room and had a microscope lab. And I was thinking about different things in natural history that visitors could do. And so I took a bunch of that clay and I sieved it. I ran it through my own screens and ended up with this sort of gritty stuff. And inside here, you can poke around under a dissection scope and find these little foraminifera. And there's all different shapes and sizes. And there's other things in there. There are in this one some ostracods and this is a piece of a really big foram. That thing was that's probably about a third to a half of it. So it was probably, pretty substantial. This gray thing here is something I use sometimes for scale in my microscope photographs. This is a piece of 0.7 millimeter pencil lead. And so it's almost a millimeter in diameter. So you can get an idea of the scale of these things. And some of them are coiled up and look like a little nautilus. And others are almost like a string of grapes, kind of. They're kind of little bulbs. And so there's quite a variety in this sample from New Jersey. And it's just getting a little bucket of clay and running it through a sieve. And these are the kinds of tools that I used, that I found handy. I would take some of that gritty stuff and put it in the Petri dish. And I would use this probe and the forceps just to move away the gritty stuff. When I found a foraminifera, I would just shovel away the other stuff and leave the foram by itself. And then use a dry paintbrush and just sort of poke it a little bit, and it would get stuck between the bristles. And the bristles would hold the foram like a little pair of forceps. and then I would move it over to a clean Petri dish and just drop it in there. And that's how I was sorting my forams. And way back in the early aughts, I did this and I made a little 4M slide where there's a little grid with numbers on it, and there's a little film on this surface that if you moisten, it becomes a little tacky, and you put the 4M there, and then when it dries, it's glued to that little box. And so you can have a little index for your sample and have a list of what's in there. Do I want to say anything else about that? No. And this is from the Navesink Formation. It's about 72 million years old or something like that. And it's a formation that does include a lot of other shell stuff. There's some oysters and some brachiopods in there. And you often run across sea urchin spines and things like that. And it's kind of a deep marine sediment. And I thought I would just wrap up. This article appeared in an online journal just about a couple of weeks ago. And it's talking about scientific voyages now, that there's a lot of the expense of running those big research vessels is insane. And so a lot of scientists are starting to hitch rides on travel ocean liners, on freighters and other vessels, and actually fishing boats. And they're sort of crowdsourcing some of this data gathering of oceanographic data on these fishing vessels. It's a very interesting article, but we've moved from Darwin being an extra on the ship, sleeping in the hammock over the chart table, to these dedicated, very expensive research expeditions. And now it's almost swinging back where, people are hitching rides on vessels that have other goals, but gathering data. There's lots of tools that can be very compact now with electronics that you could take on this vessel and just drop overboard, or, use periodically during a freighter trip, and gather some very interesting data. The article also made the point that lots of research vessels do a single straight line across the ocean, whereas a freighter might go back and forth between two specific ports many, many, many times. And so you can resample the same route and sort of see change over time in that particular profile of the ocean. So lots of interesting stuff. I'm going to stop sharing now. See if I can bump this out. I want to try to see if I can get some kind of image through one of these microscopes. Does anybody have any questions about any of these tools or the story?
Yeah, so let's